So I will go ahead and start the recording, even though we really haven't started yet, because I will forget. And I don't want to do that. So we are recording at this time, and I will continue to admit students as they come to the waiting room. And then I will introduce you oh, it is 3:30. So it is time to introduce you. And I will continue to admit students, those that are coming in a little later.
Sure.
Welcome to K-State Olathe and K-State Manhattan. This is a collaborative effort between the College of Veterinary Medicine and our campus here in Olathe to make students aware of the cases and careers in veterinary medicine. It's a lecture series that covers a variety of topics. I will put in the chat box the link so that you can, take advantage of them when you can. And I am very thrilled to have Dr. Jianfa Bai, you can correct me please, who is a clinical professor and section head of molecular research and development at the Kansas Veterinary Diagnostic Lab there in Manhattan. And we're excited to have you. And this is being recorded. So if you will hold your questions, you can put questions in the chat box, and we'll try to answer as many of those questions at the conclusion of Dr. Bai's lecture. But we are absolutely thrilled to learn about animal pathogens and those molecular diagnostic procedures that you do. Thank you, Dr. Bai.
Thank you, Masha, for the nice introduction. I'll just show my screen so that we can start. I'm hoping you see the screen now. Okay, let me just do one more thing. And do you see the screen okay? Can everybody see the screen? Thank you, thank you. So Masha asked me to talk a little bit about my career path. I wasn't prepared, but I can just briefly mention this to you. I have 3 degrees. I have a BS degree back in China, and I have a master's degree in the Philippines. So I like to travel. Then I have a PhD actually here in Manhattan, Kansas, from K-State. But my degree is from molecular biology. It's not really necessarily linked directly to animal. It's after I got a degree, and actually after I got my postdoc research experience, then I served as a service center director called the Gene Expression Facility at on the campus, and had some collaboration with VS school faculty members, and through the link, and I got the opportunity to work directly in the diagnostic lab since 2007. So it's been a while been working here. So that's basically very brief introduction about my career path. But today I'm going to talk about what we do. We are molecular diagnostic lab. We do a service with molecular assays we build. So I just want to provide some background of those technologies that is available. So for the last century, probably the most influential discovery is discovery of double helix DNA structure. So as you can see, this is a straight line. It resembles double-stranded DNA structure. And because of that, there's a list of discoveries was discovered during the last century and this century, including the three major technologies that is relevant to molecular diagnostics, which are hybridization-based. Those are like a Southern blot, RFLP, microarray, those are hybridization-based. And DNA sequencing, like a first, second, and third generation DNA sequencing, we're going to touch a little bit later on. And also PCR, I'm not sure if you heard of PCR, polymerase chain reaction. That's a major technology we've been using for routine diagnostics. Not only us, the human side, that's a major platform as well. So we been mentioned PCR already, but I'd like to introduce you about other technologies that has been used for diagnostics as well. So the first one I want to touch base is so-called Luminex. It's a magnetic beads technology. So you can have different color of beads, and those beads, our naked eye cannot differentiate, but the machine can. That's important. As long as the machine can differentiate for us, and then we can do the stuff we want. So why is that important? It's because, for example, you have 100 different colorful beads, and each one can attach to a particular pathogen, so that you can identify up to 100 different pathogens in a single reaction. But the beads is always there. How do you know it is positive or negative? Then you have another signature. It's attached to a primer we call biotinylated primer, which means you attach a biotin to the end of the primer. Then after the PCR amplification, then you wash away those those are not used primer. So only those amplified will be hybridizing to the beads. So you have two signatures here, actually, two signals here. One is the color of the beads tells you which pathogen it is, and the biotinylated primer will tell you if it's positive or negative. So the major advantage of this one is multiplexing capability. So you can detect up to 100 different pathogens in a single reaction, which is really higher throughput. However, there's no quantification. This is a plus or minus kind of testing. We're going to talk a little bit about PCR that can quantify, but this cannot. But in reality, there's a challenge in primer design, because if you add more primers are short oligos are used for amplify the target and before hybridization. So if you add more of those oligos, they start interacting to each other. So if you have an interaction we call primer dimer, then the detection efficiency drops. So there's a limitation for how many pairs of primer can you add in. That practically this technology currently can detect 2 or 3 dozen of different pathogens. So another technology called microarray, it's really hot like 20, 30 years ago. What does it do is this glass slides, 1 by 3 inch glass slides, you can put up to 1 million little spots there. This particular one only has 37,000, but the technology can allow you to put 1 million spots there or a little bit more. So what does mean is that you can put all genes of any given organism onto one slide that represent different genes. Then you harvest RNA from treated and untreated samples, then you hybridize onto the slides, you figure out which are those genes induced to a higher level by the treatment or suppressed by the treatment. So with a single experiment, you figure out all genes from the whole genome, which ones are they call upregulated or downregulated, which means induced to a high level or suppressed. Then from there, you figure out what is a mechanism of a particular organism resist to tolerate to a treatment, to an environmental factor, for example, or to pathogen infection. So it's really started with experiment for gene expression research like questions. But later on, it has been used for diagnostics as well. And everybody know the pandemic currently going on is called the SARS-CoV-2. But original SARS back then, it was also a big deal in Asia, and Dr. Wang's lab in Watch and Lewis did microarray and figured it out that this is uncharacterized coronavirus. At least we know it's a coronavirus. Then from there, people got the full genome. Then we know what the viruses looks like. So it has been used for diagnostic as well. So talking about diagnostic microarray, there's a national lab called the Lawrence Livermore National Lab, and they have a microarray covers 388 different probes, different genes or different pathogens. So it contains about 3,800 viruses, 3,800 bacteria. What do you think? It's a big number, small number? It looks like it's not too big, but however, excuse me, those pathogens are kind of included in the routine pathogens we are dealing with, including human, animal, plant, soil, whatever you can think of, the routine pathogen are here already. So it's probably the most comprehensive microarray for detection exists currently. So they said the detection limits for PCV2 and PRRS, those are animal, those are pig pathogens. They said detection limit is about CT30. That's have not introduced the PCR too much, so you probably don't know what the CT30 means. However, the detection limit for PCR is like CT35, 36. CT30 is not really sensitive, so that's a major limitation of this method. So it's really comprehensive, but detection limit is a little low. Sequencing, you probably heard of sequencing or the Human Genome Project like 20 years ago or longer. You know, for that Human Genome Project, we only have the first generation sequencing technology called Sanger sequencing that took us 13 years, spent $3 billion and in collaboration of 20 different labs. So those to get one human genome done. So with next generation sequencing, we can get it done in a week or two. So why it's so high throughput for next generation sequencing is because there are billions of reactions simultaneously occurring in the second generation sequencing, while the first generation is sequencing a single piece at a time. That's why. And for diagnostics, we call non-biased diagnostics because you can sequence everything from the sample. You probably know ATCG, that's a basic basis for DNA structure. And there's a generic nucleotide called N, and that matches all 4 nucleotides, matches ATCG. So if you use a stretch of N as a primer to amplify, then you amplify everything from the sample. So when you got a sick animal and you don't know what it is, and you can do a next-gen sequencing and figure out that way. So a major platform for second generation started with 454 parasequencing. Then very, very quickly, Illumina and mTOR and mProtein took over, because 454 is taking a picture each time you add a nucleotide. So it's a lot of storage place and processing power for computing after you got the data. When you process it, it take a lot of computing power. While the Illumina and other platforms, it's digital. So it's much easier for processing. Most people use Illumina, and some people use torrent. So that's about this. Illumina and torrent is from Thermo Fisher company. So those are the major platforms. And also in UK, there's another one coming out, and people have been really interested in this. And some people call it third generation sequencing. So you have a protein pore and supported on the solid surface. When nucleotide walking through the pore, then the electric current is altered by different nucleotide. And so that alteration in electric current can back translate into sequence. So that's the third generation. So it's really small. And also it can sequence really long piece. And the second generation is sequencing like 150, 200 bases. And this one you can sequence like 10 times more or 100 times more even. So why is that important? It's because in a lot of genome, including bacteria, they have genome rearrangement. A piece of genome is shuffled to a different place. Then with second generation sequencing, it's really difficult to figure out. But with third generation, that helps a lot. You can figure out where the reshuffling happened and where the deletion and insertion happened. However, I want to get back to first generation a little bit because we are still using it. And for I know you don't have a whole lot of knowledge of animal pathogens, but those are all pig pathogens are major pathogens, and PRRS is respiratory and reproduction pathogen. BVD is the same. Rota is mainly diarrhea. PCV are similar to PRRS and they co-occur. So we still sequence those pathogens using the first generation because people still need it. The major reason is that the first generation is cheaper and faster. The second generation, it takes like 2 weeks practically, although you can get it done in a week, but we routinely accumulate a few samples before we run it because it's way more expensive. If we don't, then we don't make any money, but so that's why first generation is still used. Give you one example is this is called the Phytogenetic Tree. It tells you which pathogen within a pathogen, which strain is similar or distantly related to other strains. So those labeled 4 strains are the PRRS vaccine strains. You can see they are all here. And it's away from here. That's why, effectiveness of PRRS vaccine is not as good. So some people use it, some people don't. So this is one example. A farm have not used a PRRS vaccine for 20 years. And what they do is to send strains to us for sequencing if they are this block and this block is, if they are similar, they won't do anything because immunity is already in the herd. But if anything different from previous soil strains, then they will dilute the fecal sample or feed back to soil. So whoever survived will be used for breeding program. So that way the piglet will be tolerant to the new strain. And so it's like you don't really produce a vaccine, but using the endogenous vaccine strategy however, using the original strain to prevent pandemic, whatever infections to newborns. So that's one example. Another example is rotavirus. Rotavirus in pigs, we have three major ones, A, B, and C. A can be cultured. So if you want to produce attenuated killed vaccine, you have to culture it first. So Rota A can be cultured, can be produced, can generate vaccine from Rota A, but B and C are difficult to culture. So it's not very easy to produce attenuated vaccine. So what people do is to sequence the VP7 segment, that is the most immunogenic unit of the virus, and stuck into a so-called vector. And they use cloned the vector to produce vaccine. And so then if the system is approved by USDA, then you have a new strain. And you put a new strain sequence into the vector, it's not regulated. So you put a new strain in, you can use right away. So that's how people still use us for first generation sequencing to get a VP7 and put it in, then he got a new vaccine. So Pointofcare, a lot of people are talking about Pointofcare, one technology called loop mediated isothermal amplification, or LAMP. I just searched the PubMed, and there are more than 600-800 peer-reviewed publications. So it's widely used. What does it do is that you do not need a lab. You do not need a sophisticated machine. You can just do it in room temperature. So the enzyme functions in room temperature. And after that, if you have UV light, the positive and negative is very clear which one is positive, or even under daylight, you can tell the difference between positive and negative. However, it requires the design of 3 pairs of primers. Sometimes it's difficult because, especially for viruses, they are changing all the time. It's difficult to find the 6th spot that is conserved among all different strains to design the primer so that they can detect majority of the strains. So that's a major limitation there. But in the recent years, there are some other technology being invented for diagnostics, Pointofcare diagnostics, called RPA, recombinant polymerase amplification. It's the same, similar to LAMP. You don't need a machine. You can do it on the farm. However, it only uses a single pair of primers, so it's much easier to design. I envision this technology probably will slowly replacing the LAMP technology. That's a few Pointofcare. I want to spend a few slides to explain PCR. Because PCR is the most used one, this polymerase chain reaction. So why is really popular and everybody is using it? It's because amplification is exponential. It's not linear amplification. So if you have 30 cycles of amplification, you got 1 billion copies from a single pair of DNA template. And sometimes we run 35 or 40 cycles, and for the cycle, you generate 1 trillion copies. So that's why it's so popular and everybody is using it. So I understand the presentation is recorded, and there is a video here tells you the basics of PCR. I'm not going to play it because I'm not sure how it works. But later on, if you want to watch it, it's a good source to understand the basics of PCR. So real-time PCR is really a PCR machine. Then you have a digital camera on top of a PCR machine to monitor the signal from each cycle of amplification. So for example, you have a template they diluted 10 times, then you can tell exactly which concentration is for each dilution. However, for regular PCR, we call traditional PCR, regular PCR, or conventional PCR, then you have to run on gel, agarose gel, then you can tell that they are positive, but you have no idea of original concentration. So there are two advantages, major advantages for real-time PCR. One, you can quantify the original concentration. Two, it's more sensitive. It's not because of amplification, but because the detection technology for real-time is more sensitive than regular. Also, both can be multiplexed. For real-time, you label the probe with different dye, so you can multiplex them, but it's up to 5 colors. That's what the machine is available commercially currently on the market. You can only buy machine for 5 colors. There's only one system that can detect 6 colors. But for regular PCR, if you can design different genes with different molecular weight, then when you run on gel, they can be separated. Then you can put more pathogens or genes on one reaction. So that's how you multiplex them. The detection chemistry, you have a long list, but most people do Cyber and Pagma. I'll just touch base of these two. The Cyber is kind of a double-strand DNA intercalating dye. So it cut into double-strand DNA and generates signal. And the major issue here, not really issue, but something you need to know is that the cybergram is not specific. It will generate signal for any double-strand DNA. So you need to watch contamination if you use Cyber. The Pagman is a one, you have a probe binding to the place between the primers. Those are primers, so amplifies. Then you have a probe with a quencher next to it. So when the probe is intact, the quencher absorbs the energy from fluorophore and transform into heat, so you don't see the signal. Then the DNA polymerase comes in and chops the fluorophore out. Then the fluorophore is away from quencher, then we see signal. So the major advantage here is that the signal is generated only during amplification. So no amplification, there's no signal. So we are molecular R&D. We kind of are building molecular assays all the time. But one question we were asked is why? Why do you need to do this? Because when you look at the literature, there are tons of literature there. Why don't you pick one and use it? So this one example, I try to explain why we used to design the primer probes ourselves, like this one in particular. You have a reverse primer that coverage is 60%, so you are missing 40% of the strands. That's not good. And a few years ago, we reanalyzed, and it's getting worse. You detect 40%. So that's why not every published protocol can be used for diagnostic directly. Another example I like to mention is this swan flu sample. We have 23 virus isolation positive. Virus isolation is gold standard for traditional virology detection. So they are truly positive, but we have missed quite a few by qPCR, qPCR, real-time PCR. So we sequence them, and the yellow highlighted are the primary probe, and not highlighted are sample. So the probe is really good, and forward primer is good, and reverse primers have two. Both are missing, have some mutations as indicated in red. So that's why PCR is not working. So, but this assay was developed by USDA experts. We know they are good, and they know how to do this. So why we are still missing strength. It's because the viruses are changing all the time. So when you design an assay, it covers like 100%. Few years later, you have new strains emerged, then your assay is no longer covering the new strains. So it's a continuous process for molecular assay design and monitoring. It's not a one-time deal. So that's why we kind of just do stuff ourselves. So what is the current trends for PCR? It's still the major platform everybody is using, we know it. And people started doing a syndromic panel testing and including internal controls. So I'm going to use one example. This is a bovine respiratory panel. So you have a list of pathogens and a list of bacteria and viruses that is a common pathogen to bovine respiratory tract. So if you do one at a time, it'll take maybe 2 weeks to figure out what is the causing agent of the disease. If you do a panel, you run everything at one time, then you figure out the next day. So that's why it's very helpful. Internal control is the one, you try to monitor, make sure your extraction and PCR are good. So you detect a pathogen, then if it's positive, that's everything is good. But if it's negative, how do you know it's true negative? Maybe you have a extraction failure. But any kind of a sample except the environmental sample, like a blood, serum, tissue, fecal, nasal, oral, all samples have a tons of host animal cells in it. So if we design primer amplify a host gene, that can be used as control. So give you one example here. This is canine brucellosis case. So for those positive ones, it's pretty good. For those negative ones, then the control is working, generate signal. So you know it's true negative. So you don't worry about it. You just send data out. This is a summary of internal control. I think it's a little bit too much. We don't need to talk too much. Each have advantages and limitations. If you want to know more, we can talk more, but it's a little bit too much for this talk, I guess. I'll just walk through the basic steps of how to build a molecular assay a little bit. So you select a target, and you got all sequences, and you do sequence alignment, and you design primer probe that is conserved within the pathogen, but it's a specific across different pathogens. So it specifically detect the majority of the pathogen in your target species. Then you do so-called analytical validation. So you have a high concentration of a strain, then you do 1 to 10 dilution and see how it works, if the standard curve is linear or not. So that's R square, called correlation coefficients. If it's greater than 99.99, it's good. And E is amplification efficiency. If it's greater, if it's in between 90 to 110%, then it's good. Then after that, you do diagnostic validation. Really, you just use a real diagnostic sample to figure out diagnostic sensitivity, specificity, repeatability, and reproducibility, and verify what kind of sample should be used for the assay. And the AAVLD is a society that certifies diagnostic labs in the US and Canada, and they recommend to test 30 positive and 30 negatives before you provide the service. So we are following their suggestions in general. And there's one practical question for real-time PCR That's sensitivity level. We've been talking about PCR is so good and so sensitive, but in practice, we need about 5,000 to 10,000 copies to get. So why is that? It's because when you do RNA, DNA extraction, you started with about 100 microliter, that's 1/10 of a ml. And when you elute, you don't use up or elution for PCR, you only use a proportion of extracted nucleic acid for PCR, because if you overuse, it still have inhibitions. So really, 5,000 copy per ml is equivalent to 5 copies per PCR reaction. So that's why we still need 5,000 to 10,000 copies for PCR to work. Another question we've been asked really routinely is pooling. Can we pool or not? The answer is you can, but in this particular case, you are going to miss 6 to 7% of positives. So it's really depends on what you want to do to save some money or to be more accurate. Sometimes just the screening, if you pull, that's fun, but sometimes you want to find out which exactly animal is caused by which pathogens and you don't want to pull. So that's kind of, so diagnostic lab, just very briefly, we have all different sections listed here, also molecular, but in molecular, we also have 3 different sections. My section is molecular R&D, so build assays and validate them. After validation, we move to Dr. No's molecular service lab to provide service. We also have a sequencing lab for next generation sequencing. So if you are interested, on the website, we have a portal here. If you put the PCR there, you are going to end up with about 120 different assays we are providing at a routine basis. Also, there's a button called More if you want to find out more information. Then it'll provide you more information for a particular assay you are interested. For example, this particular canine parvovirus PCR differentiate 2A, 2B, and 2C strains, and they tell you what kind of sample you need to submit and what is the cost, etc. And also, if you want to put the sequencing as a keyword in the search, then you have a list of 1st and 2nd generation sequencing tests that is available on the website. I guess that's pretty much I want to cover for technology and what we've been providing, but I also want to touch base a little bit about COVID-19, since it's something people are kind of concerned of. We call COVID-19 all the time, but that's the disease name. The virus is called SARS-CoV-2. So the disease name and virus name are different. As of today, this morning, I searched the WHO website. We have 228 million confirmed human infections and over 4.6 million deaths globally. So it's really a terrible pandemic. In relation to animal, the human receptor called ACE2 receptor means the binding component to the virus. The virus find the ACE2 and binding into it to gain entry into human or animal cells. So the receptor gene is only 81, 84% to the dog, cats, cattle, and pig genes. Those are common animal we're dealing with every day, but it's really almost identical to non-human primates. So routinely, if you're dealing with dog, cats, and cattle, and pigs, it's not a major concern. There's no evidence that those animals is transmitting the disease back to human. It's probably the other way around. So kind of human passing the virus to animals. Currently, we have 234 total positive animal cases in the US, exclude the mink farms. So just other, very recently, last couple of weeks, we got white-tailed deer infected, and it's confirmed positive. So really, we are concerned is the variants in the beginning, the UK variants, the South African variants, Brazilian variants, and now most people are concerned of Indian variants. The major characteristic N501Y means the amino acid N at 501 location is mutated to Y, etc. And this one is deletion, of course. So that labels different variants. Currently in the US, vast majority of infections caused by the Delta variants. So, you know, people have been arguing a lot about vaccine stuff. And, you know, people have been wondering if vaccines still work or not. About this news called a news break I read is indicating this is from CDC data, 99.999% of vaccinated people do not end up in hospital and still survived. Other side of data indicating those hospitalized and those did not survive, dead people are not vaccinated. So it correlates really well to each other. So when you look at this CDC data here, sorry, I should take out this one first. So the Johnson County is one of the county in Kansas that have the highest vaccination rate, 74.6. And this is today's data. This morning I got this data. And really surprised to see Riley County, where K-State is located. We only got 37.3% of people fully vaccinated. So that's a little bit bizarre. In most college town, you have higher rate of vaccination, but this is not the case. And actually, it's close to the bottom percentage of vaccination in Kansas. I want to show you just one line of data. This morning, we got a positive case that have a CT of 13 or 14. You probably don't understand, if you haven't done too much PCR, you probably don't know what does that mean. It means the person have 8 million viruses per mil. That's per mil is per, yeah, when you extract it's per mil, but it's really just per swab. So when you swab it, you put into a mil of buffer, and that's a mil. So one swab, you got 8 million viruses. That's tons of viruses there contaminating other people. So that's really a very high concentration we have been seeing. And everybody probably know president have an executive order require all federal employees to get vaccinated. And K-State people, we are still evaluating the situation, see if we should adopt the regulation as well, but we have not so far. So the key thing here is really to get vaccinated. If people everybody get the vaccine done, then the viruses will not have the chance to mutate. If we don't get enough people vaccinated, then some people got sick. Then in sick people, the viruses keep mutating. That's a problem. If everybody got vaccinated, then the virus will not have the chance to mutate, then that can be the end of the pandemic. I understand I probably did not cover enough that you are interested, particularly I probably talk too much technical stuff that may confuse you. But I do have my email address here in case you have questions later. But if you have some questions you want to address now, I'll try my best to answer. Thank you for your time.